predisposed mice who were subjected to a stroke, and that this dysregulation actually led to larger infarct sizes. And it's really interesting that later on they showed indeed that these primed immune cells in the gut were able to translocate into systemic circulation and exert their effects, their dampening effects within the brain, which really implies that there is a gut-brain axis in the context of stroke. Other studies have also shown that the microbiota is able to produce specific metabolites. We talked about short-chain fatty acids and secondary bile acids, which are also immediately able to translocate from the gut into the systemic, but also the lymphatic circulation, cross the blood-brain barrier, and also at a beneficial system in the acute phase of stroke. And there's also less well studied, but hypothetical knowledge that shows that the enteric nervous system and the vagal nerve could also be implicated in reducing stroke infarct size and improve outcomes, which also make it very interesting that the microbiota could be is a modifiable target that can improve infarct and stroke outcomes. My interest, of course, is in infectious diseases, so I would be remiss to not talk about the role of the microbiome and post-stroke infections. But it's also very important in the context of stroke patients. And a study over a decade ago by one of the moderators has shown that in over 130,000 patients of stroke, 30% of those patients had infections in the first days following hospital admission. 10% of those patients had pneumonia, and they showed that pneumonia in itself was an independent predictor of mortality in those patients, which means preventing pneumonia in those patients is very important to improve outcomes. Why do stroke patients have an increased risk of infections? Well, the risk could be explained threefold. The most logical explanation could be that these stroke patients suffer from partial paralysis, making it more difficult for them to swallow, risk of aspiration pneumonia. Simultaneously, it's been shown that stroke itself causes severe immunosuppression, making these patients more susceptible to infections. And in addition, it's also been shown that the gut in stroke patients loses its integrity, and there's an increased risk of translocation of these gram-negative enterobacteriaceae causing infections by itself. And specifically, those latter two risk factors could be potentially modifiable by the microbiome, and specific the anaerobic butyrate-producing bacteria. Because butyrate in itself and short-chain fatty acids have been quite well studied and are playing important roles in protection against infections. On the local effect, butyrate has been shown to be the most important energy fuel for colonocytes, leading to increased intestinal epithelial integrity. But simultaneously, it's been shown that butyrate is able to translocate from the gut into the systemic circulation and prime both the adaptive and the innate immune system. This is an example done by Schultes in macrophages that's shown that butyrate, which is most often produced by one important obligate anaerobe that's been discussed at length already this session, Faecalibacterium parsonitzii. Butyrate is recognized by G-protein coupled receptors, GPR41, 43 to be exact, which leads to epigenetic remodeling of your immune system by inhibition of HDAC3 and reduced glycolysis, reduced mTOR signaling, and an increased antibacterial activity of these immune cells. But simultaneously with this increased antimicrobial activity, there's also more of a dampening immune effect. There's increased IL-10 production, but also an increased amount of T regulatory cells. So it's really interesting that these cells are better capable of attacking infections, but simultaneously they do not cause any collateral damage to the immune system or the tissues surrounding it, making the microbiota really interesting modulators of our immune system and protection against infections. So we went back to our cohort of 349 stroke patients and assessed their butyrate-producing bacteria abundance, and showed indeed that the presence of these butyrate-producing bacteria was an independent protective risk and protective effect to reduce the amount of infections. About a 25% decrease in infections were seen in patients with enough amount of butyrate-producing bacteria in their system. And we looked specifically at Faecalibacterium parsonitzii in this context, and showed that indeed there was reduced amounts of Faecalibacterium parsonitzii in those patients who had stroke, and reduced amount of Faecalibacterium parsonitzii in patients who developed stroke infections. And simultaneously, we also, if you had enough Faecalibacterium parsonitzii in your gut, you were increased, more quickly successfully discharged compared to those who had lesser amount of Faecalibacterium parsonitzii, making this specific bacteria very interesting for next generation probiotics. But it's also an obligate anaerobe, very susceptible to oxygen, making it very difficult to get it to come where it's needed. So other tools are needed, and I'll talk a little about that in a couple minutes. So throughout the potential events towards stroke, from susceptibility to outcomes to infections, there are potential avenues for which the microbiota could be of importance. So I've created a schematic graph to show just one patient and its microbiome and how this could be modified in the potential phases towards stroke. And it starts with someone who is susceptible to stroke, for example, someone who has hypertension, hypercholesterolemia. And because of some trigger, antibiotics, that you start having too many hamburgers, you lose some kind of your microbiota diversity, and it also introduces these gram-negative Enterobacteriaceae who start producing more TMA and causing actually that positive feedback loop, which actually enables the microbiota to change and to increase the amount of TMAotas in your system and hence increase your risk of stroke. The moment a patient is hospitalized with a stroke, we also see that there's an even starker drop in your microbiota diversity with even more Enterobacteriaceae, making you more susceptible to opportunistic infections. I haven't much told about the long path to recovery for many stroke patients, but you can also imagine that the microbiota after all these insults will try to slowly recover. And all these three phases, pre-stroke, during the acute phase of stroke and post-stroke, you can envision all kinds of interventions that can be employed to modulate the course and potentially improve outcomes. You can think of probiotics, symbiotics, maybe combining with prebiotics, but also postbiotics, giving them the short-chain fatty acids, or employing fecal microbiota transplantations. But I think it's very important to realize that very often the probiotics that have been employed are non-specific to the effects you want to establish. And it's already really eloquently discussed in the first talk. I think you really have to understand the mechanisms that are at hand before you give just blindly the probiotic that you can find off the shelf of a supermarket. And I think that's where the studies or the knowledge that we have is severely lacking. But there's a potential future, which is actually quite promising, because the group of Stanley Hazen actually identified an inhibitor of the converting enzymes that convert choline and carnitine into trimethylamine. And it showed that in mice with susceptibility to become obese, that when they give this inhibitor, these mice were actually retaining their weight quite well, did not get obese, and also had reduced amounts of cardiovascular events. And most importantly, they were able to eat the same amount of foods as much as they wanted while retaining their weight. It's kind of like good to be true, which means probably I could eat twice as many tortillas as many as I don't want to do. But also, on the other hand, in terms of next generation probiotics, there's a recent study that came out from a group of Frederick Beckhead in Göteborg that showed that Faecalibacterium parsnitzii, which again is very oxygen sensitive, through genomic adaptation, they can make it a little bit more tolerant to oxygen. And then when they tested it and gave these new oxygen-tolerant Faecalibacterium parsnitzii to human volunteers, they identified the Faecalibacterium actually in the gut, which meant actually that you can envision novel next-generation engineered probiotics that potentially have effects and also understanding mechanisms as to why they potentially improve outcomes. So I think there's definitely some important future prospects that could definitely modulate the risks and outcomes of stroke patients. So my conclusions are, hence, that the microbiome is linked to the pathophysiology of stroke, both from an increased risk to stroke as well as altered outcomes. I think the preclinical models, the mouse work is very compelling, which I think is many microbiota studies, but the human data is associative and sparse, which I think is a challenge that we need to address as a group across disease disorders to try to tackle. Because I think if you also hear the talks that we had, we do see similar pathways, similar bacteria pop up. So we need to think about the functions and see if we can identify tools that we can broadly distribute across these disease types. And I think that the research community can be better involved in this. Simultaneously, I think our ability to modulate the microbiome gets increasingly more sophisticated. Our sequencing tools are getting better. We can almost now sequence to strain level resolution. So I have also strain level functional capability of why certain bacteria work and others don't. So I really think that the time to start better informed trials with next generation probiotics now. So I'm very happy to see where we'll be in the next 5 years, because it's exciting times ahead. Many thanks to many people who've contributed to this work, both from Amsterdam UMC, some of the people are in the room or even moderating, and also many thanks to my current employer at the Serum Institute, who've helped me learn more about microbiome and potential therapeutic applications. I'm very happy to take any questions. Thank you.